I'm Miss LaGraves, and we're starting Unit 2. So, Unit 2, cell structure and function. We are looking at the big deal here. Cells are a basic unit of life. They provide environment for organelles to function, have a membrane to maintain homeostasis. So that's sort of the overall arching question here. Today we're going to do topics 1, which is cell structure, subcellular components. You can see your vocab here, your essential knowledge. We're doing topic 2, cell structure and function. You can see your vocab over here, essential knowledge over here. It looks like a lot, but it's kind of going to wrap up nicely. We're also going to be doing topics 10 and 11 today. So compartmentalization and origins of cell compartmentalization. I just feel that they fit together really nicely with the idea of subcellular organelles and structure and function. So you can see objectives and essential knowledge over here. So you're going to start your notes here on page 6. There are some reviews and some free response questions we're going to skip. Those are on pages 8, 11, 12, and 16. I'll tell you when those are skipped. So I also have here the 3D model that we were working with in class, taking out the organelles, and labeling them. So you can use this just as a reference. This link is still on Classrooms. So feel free to use that as a reference as you're listening to the notes. Great. Let's get started. So this unit, cell structure and function, we're going to talk about subcellular. Okay, so there's cells and then sub on the inside of cells. So these are the components inside of cells and how they sort of compartmentalize. So cells, as we should know, this is the basic structural and functional units of every organism. There are some key features that they have. The first is that all cells are bound by a plasma membrane, which means they all are, this is my water balloon analogy, that cells are all kind of contained like a water balloon is, and the balloon is the plasma membrane. All cells contain cytosol, which is the fluid environment that these organelles live in. They also contain chromosomes, which is the inheritable genetic material that is passed down. And all cells contain ribosomes. That's an important one. The function of ribosomes is to create proteins and other things, so we'll get into that. But these are 4 features that all cells have. There's 2 types of cells. There's prokaryotes and eukaryotes. Prokaryotes are things like bacteria and archaea, right? They're the small, simple ones. Their DNA is in a nucleoid region. So if you look at the cell on the 3D model that you have, you'll notice that there's a nuclear envelope around the DNA. Prokaryotes don't have that. Their DNA is kind of just floating around in the cytosol, and they're generally smaller than eukaryotes. Eukaryotes I like to think of because you are a eukaryote. So protists, fungi, animals, plants, all the things that in our minds we're thinking like, yeah, that's alive. You know, animals, plants, you're a eukaryote. In eukaryotic cells, DNA is in the nucleus, so it's bound by that membrane, and it contains other membrane-bound organelles as well. Everything else is kind of a water balloon inside of a water balloon. So let's get into these organelles. Organelles, what they are is a membrane-bound structure in eukaryotes, particularly in eukaryotes. There's 2 classifications. We have endomembrane organelles, meaning endo meaning inside, membrane meaning membrane. These organelles are bound inside of a membrane. There's the nuclear envelope. There's the endoplasmic reticulum. There's the Golgi complex. There are lysosomes, vesicles and vacuoles, plasma membrane. All right, let's go over each one of these. I probably should have gone over each one of these to begin with. So the nuclear envelope is what we consider to be like the nucleus, right? When we look at it under a microscope, it's essentially the structure that surrounds our DNA, right? Endoplasmic reticulum is found right outside the nucleus, or the nuclear envelope, I should say. And its function is to sort of receive the mRNA or the DNA from the nucleus itself. Golgi complex kind of packages it up. Lysosomes take out the gross stuff and all the trash. Vesicles and vacuoles are just like what they sound like. My water bottle is a vesicle for water. Like they take things in. Plasma membranes obviously hold everything together. Then we have our energy organelles. Mitochondria is found in animal cells as well as plant cells, and these produce ATP. ATP is they call it the energy currency of the cell, but it's essentially like a molecule that produces energy that allows these cells to go through this biological process. Like, we can't have a reaction take place without energy, and the mitochondria is the source of that energy. That's why they call it the powerhouse of the cell. And then chloroplasts. Chloroplasts are found only in plant and photosynthetic algae. We'll get into it, but it's the green stuff in plants that takes in light and photosynthesizes. Pretty cool. All right, let's talk about compartmentalization. So if I was to ask you to like compartmentalize things, like we're going to put them in different boxes separate from each other, right? Compartmentalization in organelles, what it does is it allows these reactions to occur in different locations. So I can have something going on in the mitochondria that has nothing to do with what's going on in the Golgi, and like, that's great. We're all doing our own thing. It also increases surface area, for reactions to occur. We'll look at that as well. If everything's kind of compartmentalized, you can have different foldings of membranes on the inside and on the outside to increase surface area. And really, in this unit, increasing surface area is the name of the game. We're going to have a big lab on this for topic 3. Great. It also prevents interfering reactions from occurring in the same location. In terms of like mitochondria, right? So we have the inner and the outer. We can have these reactions without interfering with each other in the same location. Pretty cool stuff. So there are a couple differences between plant and animal cells. In plants, they have exclusively chloroplasts. This is plants and photosynthetic algae. Excuse me. They also have a central vacuole. It's right over here. It's really big. It holds water. It increases turgor pressure. We'll get into it. So like, it keeps the plants kind of upright and rigid. They have a cell wall. It's outside of the cell membrane. And they have these things, plasmodesmata, right? These are these little pores outside the cell wall. In animals, we have lysosomes. Let me see if I can find a wee little lysosome here. I think these tiny little ones, they're all over the place, but they have hydrolytic enzymes in it that just break things down. They're like the waste collectors. We also have centrosomes. Centrosomes are these guys, and they aid in DNA replication. Some animal cells have flagella, which are these weird tails, right? Also, they have pili? No, these little things, the short things that look like hair, those are called pili, P-I-L-L-I. We also have those in animal cells. Cool. Endomembrane organelles. So these are the organelles in eukaryotic cells, in your cells, right, in animal cells. We're going to go over each individually. First is the nucleus. Nucleus contains chromosomes. Chromosomes are giant long pieces of DNA that hold genetic information. This is what is passed down from generation to generation. They are enclosed by a nuclear envelope. You can see here it's double membrane. So you have an inner and an outer membrane. That nuclear envelope has pores. You can see them here. Pores are just tiny little holes that things can go in and out of. They regulate entry and exit of materials from the cell. So the materials that are inside of here is the chromosomes. The things on the outside of here is the rest of the cell. So things have to pass in and out, and it's the job of the pores to be the gatekeepers of that, so to speak. Oh, there you go, pores, boom. All right, nucleus. It contains a nucleolus, right? So this whole thing is your nucleus. If you can imagine, there's an even larger cell. Ooh, that's a weird looking cell. Larger cell around it with other things going on out here. And this is your nucleus. Inside the nucleus is your nucleolus, right? Really dense kind of circle that you'll see under a microscope. It's a dense region of the nucleus where rRNA is synthesized. rRNA combined with a couple proteins actually make up ribosomes. So this is kind of an interesting concept, right? So like, where do these organelles come from? Like, where are they coded in our DNA? And it is sort of its own DNA, right? So there's a particular part of your DNA codes for this ribosomal RNA in order to make protein that can code DNA. It's very strange, but quite brilliant. Awesome. Subunits exit the nuclear pores. All right, so it's made in here, and they sort of assemble, and then we go outside. Awesome, to do like our job. That's your nucleolus. These proteins, these rRNA and proteins, these form ribosomes. All right, so these ribosomes are made somewhere. They're made in the nucleus. Ribosome's job is to translate messages found on mRNA. So first comes the rRNA plus the proteins. They leave, and we are able to make proteins out of other mRNA structures. This is going to be wicked important when we come to unit 6. But this is part of what we call the central dogma of science. We'll get into it. All right, ribosomes. Speaking of ribosomes that are made in the nucleolus and combined with proteins to create ribosomes, they are combined of ribosomal RNA and proteins. We just talked about that ad nauseum. Their function is to synthesize proteins. It's like, what came first, the ribosome or the protein? Some texts don't classify them as organelles. We are going to classify them as organelles because their job is so important. They can be found in two places: free roaming in the cytosol, right? So remember, the cytosol is the fluid stuff that everything is in. Proteins produced here generally function within the cytosol. So proteins are they have a lot of functions in the cell, and one of them is enzymes. Enzymes speed up reactions. We'll see that in the next unit. We actually have a whole unit on it. So buckle up, we'll get there. But proteins that are hanging out in the cytosol remain in the cytosol. Then we have, we call them free ribosomes. They're free range. They're free range ribosomes. Then we have ribosomes bound to the endoplasmic reticulum, the ER, or the nuclear envelope. Proteins that are produced here generally go out of the cell. Okay, so produce something, send it out. And they leave via a transport vesicle. It's essentially just a membrane-bound vesicle that takes these proteins and escorts them out of the cell. Um, Great. There you go. There's your free ribosomes. There's your endoplasmic reticulum. It's that funky looking thing. Awesome. Speaking of the endoplasmic reticulum, what it is that a network of membranous sacs and tubes. They do a couple things. So I'm sorry, I just wanted to dump that slide on you. This was my analogy with the water balloons. This is like the balloon animal balloons filled with water stacked on top of each other, right? It's a network of membranous sacs. It synthesizes membranes on the rough ER. And the compartmentalization of this is to keep proteins formed in the rough ER separate from the free ribosomes. So we want to make sure that the proteins that are produced on the rough ER make it to the place that they're supposed to, as opposed to the ribosomes that are producing the proteins that are free range, free ribosomes. Got it? Cool. All right, yeah, two types of endoplasmic reticulum, rough and smooth. The rough ER has those ribosomes bound to it. So you can see that here. It's the dots. As far as the cell goes, we're right about here. So this is your nucleus, double membrane. That's your nucleolus. You can even see the pores. And we are right here and right here. So the endoplasmic reticulums are very close to the nucleus. Makes sense. The ribosomes have to take the genetic material and translate it. So it contains ribosomes bound to the endoplasmic reticulum membrane. You can see that here. Smooth ER doesn't have any ribosomes. The purpose of the smooth ER is to synthesize lipids. It metabolizes carbs, and it detoxifies the cell. So lipids go into the membrane. Carbohydrates are used for energy and glucose storage, and it also sort of traps these toxins from the cell and transports them out. Great. Golgi complex, yay! Golgi complex is similar in to the endoplasmic reticulum in the sense that it's like these flattened membranous sacs. You can see them here. The big difference is that these sacs are not connected to each other. They're called cisternae. They're not connected to each other. They're separate. They separate the sacs from the cytosol. Cisternae are not connected. So here we go. One of these little weird kind of balloon things. That's called the cisternae, and each one is not connected. And it has directionality. You have a cis face that receives vesicles from the endoplasmic reticulum, and you have a trans face. It sends the vesicles back out into the cytosol. So you can think of the Golgi complex as the mail room, right? Or the UPS. It receives stuff from the ER, and it packages it, and then sends it out, right? So there's directionality to it. Cis face receives, trans face sends. Boop, boop. Its functions, as I was just saying, it receives transport vesicles with materials from the ER. It modifies the materials if it has to be modified, and it sorts them into vesicles to send them out. It also adds tags to things, right? Again, it's like the mailroom. So a piece of material comes in that's meant to go out of the cell as a communication signal. So it can add that tag and get it sorted to the right place. Packages materials into new transport vesicles, and those vesicles exit the membrane via a process called exocytosis. Exo meaning out. Cytosis meaning cell. So out of the cell, exocytosis. These modified materials, as I said, they ensure that these proteins are folded correctly, modified correctly, and gets out to the right place. Cool. Lysosomes. Lysosomes are pretty cool. I actually really enjoy. I've grown to appreciate lysosomes in my old age. They're these just membranous sacs, right? So it has this double membrane. Once again, you can see the outer and the inner. And inside of it, they have hydrolytic enzymes. Their function is to hydrolyze macromolecules. So they break down these macromolecules. Remember the hydrolytic process? Breaks down macromolecules. Unit 1's coming back to haunt us. It does this process by this term autophagy. Auto meaning to oneself, phagy meaning to eat. So they recycle their own cells' organic material, right? So they're like the trash collectors. They're the recyclers. It allows the cells to renew themselves and like take out the trash. So if a process goes down and it breaks it down, now we have some excess materials. Through this process of autophagy, these lysosomes can recycle it down to its basic components and we can reuse it. Peroxisomes, I mentioned here just because they're kind of similar to lysosomes. They catalyze reactions with hydrogen peroxide. So they produce H2O2. And then, yeah, that's it. We're going to do a lab on peroxisomes. But remember peroxisomes because peroxide produces hydrogen peroxide, per meaning two, oxide meaning two. Anyway, now we got vacuoles. They're super large. They're found only in plants. There's a couple different types. So we have a food vacuole. This is formed by cell eating, phagocytosis. Phago meaning to eat, cytosis meaning cell. And then they're digested by lysosomes. They also have another type of vacuole called a contractile vacuole. This maintains water levels in cells. And the central vacuole found in plants, this contains inorganic ions and water, and it's important for turgor pressure. So I went over that really fast. So vesicles, they're essentially, or vacuoles, they're essentially just like, they're like the bags of the cell. Like we have to put something into a bag, here's a vacuole. You can store food, it can store water, and central vacuoles in plants are really big. That's your central vacuole. And if anyone has a green thumb and has noticed, if you don't water your plants for a while, they kind of get a little withered. They're like, no. It's because this central vacuole, the water in it, is being used to like keep the plant alive. And then all of a sudden you water it, and like 10 hours later, not even 10 hours later, it's like, ooh, I'm perky again. It's because the central vacuole is like getting fat, right? With water. It's pressure, the pressure of it kind of plumps things up. What we're seeing there is what we call turgor pressure. It's also important to kind of keep that rigidity in plants. So as that water is, pushed into those vacuoles, or osmosis occurs that allows those vacuoles to get bigger, the plant kind of perks up. Interesting stuff. Boom, there it is. Sorry. Let me get rid of all my nonsense. All right. So we just went over 6 organelles. Let's talk about the ones that give everything else energy. So this is topic 211. We're going to talk a little bit about endosymbiotic theory. This is a theory that explains the similarities between mitochondria and chloroplasts and the similarities they have to a prokaryote. So the theory states that an early eukaryotic cell, so this is kind of how we went from like a simple prokaryotic, this is like billions of years ago we're talking, it engulfed another prokaryotic cell. And that prokaryotic cell became endosymbiont. Endo meaning inside, symbiont meaning like relationship, a cell that lives within inside of another cell. It's like a little parasite inside of it, but like not, because we're just talking about cells. They became able to function together, right? So one prokaryote, really simple cell, came along billions of years ago, ate another prokaryotic cell, and the prokaryotic cell inside the other one was like, hey, I can give you something, produce some energy. You mind if you carry me around for a while? And they were like, yeah. And then they became one functional organism. There's some evidence to this, right? And it's pretty, we can like see this with our eyes. So double membrane system. So the fact that there was something and then like something else ate it. And then you can see now there's a double membrane on the inside. Like it's weird, right? Ribosomes, the fact that they're like free range, they make their own proteins, circular DNA. So we're going to get into this for real, but circular DNA. Capable of functioning on their own. So mitochondria, chloroplasts, some of them are able to function on their own. And again, mitochondria has their own DNA. We're going to talk about that one as well. That's like a wild, wild thing. Speaking of, let's talk about the mitochondria. Mitochondria is the site of cellular respiration. The structure itself is that double membrane system. So you have an outer membrane that's smooth. There's your outer membrane. And then you have an inner membrane called cristae. They are like these weird kind of wavy things. It divides the mitochondria into two internal compartments. So you have one here and one here. And those two compartments are totally separate from one another. And what this does is it increases surface area. Surface area is the name of the game in this unit. Increase surface area, decrease surface area. We're going to have a whole lab about it. All right, so in the inner membrane is the space between the inner and outer membrane. It's like the one time science makes sense. Intermembrane, the space between the inner and outer. And then you have the mitochondrial matrix. So you got this is the inner membrane. Then you got the matrix in over here. The location of the Krebs cycle occurs here. That's kind of all you really need to know about it at the moment. We're going to get into the Krebs cycle in detail in Unit 3. But for now, just know that it takes place in the mitochondria. The mitochondrial matrix also contains enzymes that catalyze cellular respiration. So enzymes, the function of them is to speed up reactions. Catalyze means to speed up. So we speed up cellular respiration, and it produces this molecule, ATP. As much as surface area is the name of the game in Unit 2, ATP is going to be the name of the game in Unit 3. It also has mitochondrial DNA, which is fascinating. We're going to talk about the mitochondrial DNA. It's separate from the DNA in your nucleus. So you have two sets of DNA. Haha, did no one tell you? Well, now we know. And it also has its own ribosomes. So this structure here is really kind of a piece of evidence in the endosymbiotic theory. It kind of feels like that with its own DNA and its own ability to produce enzymes and its own ability to produce its own energy. I don't know, it could have been its own cell at one point, eaten by another cell. There you go, inner, boom. So the number of mitochondria in a cell correlates with its metabolic activity. So the more energy we need, the more mitochondria the cell will have. Your muscle cells are lousy with mitochondria. They constantly need energy to be pumping and stretching. Chloroplasts, specialized organelles in photosynthetic organisms. So it is the site of photosynthesis. Light comes in, whole bunch of reactions happen, energy comes out. It contains this green pigment, chlorophyll. It is not the plants or the leaves that make it green. It is the chlorophyll that makes it green. Inside of this double membrane here, you have these things called thylakoids. Thylakoids are membranous sacs, so again, bound in a membrane. If we see membranous, that means it's a balloon. Sacs that can organize into stacks called grana. So light-dependent reactions occur here. So we can see, let me just clear this. There's your thylakoids. There's your grana. Little pancake structures are called grana. Not grandma, grana. The stroma is the fluid around the thylakoids, right? So similar to the cytosol in cells, the stroma is the fluid around it. It contains chloroplast DNA. So plants also have separate DNA that is separate from their nucleus. 2 sets. Well, actually, I shouldn't say that. I don't know if the plants have 2 sets. Eukaryotes do, though. They also contain ribosomes, which support the endosymbiotic theory that the chloroplast and the mitochondria were at one point their own prokaryotic cell. They contain enzymes in order to catalyze reactions. So there you go. Proof of the endosymbiotic theory. I shouldn't say proof. There's no proof. Supporting fact. All right. Let's talk about the big bones of all of this, right? Like how do all these organelles exist in this weird kind of balloon? It's, we call it the cytoskeleton. So it's a network of fibers throughout the cytoplasm. So cells are, there's a lot of liquid. We talk about the cytosol being liquid, but within that, there's this like scaffolding of fibers. It gives structural support, especially for animal cells, because we don't have a cell wall, and mechanical support. So it can anchor the organelles in place, and it can actually, it's like this little network of like train tracks that go through the cell that allow for the movement of vesicles or organelles or the whole cell itself, right? So little train tracks, little scaffolding. The movement occurs when the cytoskeleton interacts with these things called motor proteins, and they're exactly what they sound like. They have a motor on them. They kind of move things around. There's 3 different types of fibers found in the cytoskeleton, and they all do different things. You have microtubules, which super small, tubules, it's a tube. It's like a straw. Micro, super teeny tiny filaments. Filaments are solid. They have more structural support. They're not hollow like straws. They're solid like a baseball bat or a 2x4. And then there's intermediate filaments. They kind of have their own jobs. We'll get into it. Just know they exist for now. Microtubules, so hollow rod-like structures built of a protein tubulin. Oh, thank goodness. Tubulin is in microtubules. Tubulin is the protein that makes microtubules. Tubulin. They grow from the centromere. That is that little organelle that exists primarily in eukaryotic cells. They assist in microtubule assembly. That's what the centromere does. So you have these things to anchor off, and then the microtubules made of tubulin kind of branch off of it. But this is the centrosome. It's the thing that it anchors off of. There's a bunch of different functions. These serve as structural support, right? Think, again, tracks for the movement of organelles that are interacting with motor proteins. So the tubules are the things that move the organelles, move the vesicles around. They assist in the separation of chromosomes during cell division. So when we get into mitosis, meiosis, when we talk about cell division, microtubules are the things that grab on and pull to either side. And they assist in cell motility, right? Motility, moving around, like cilia or flagella, the weird tail that hangs off of things. Microfilaments. So microfilaments are solid rods made of a protein, actin. I'm sorry that it doesn't work as well as microtubules and tubulin. I wish it did. They function to maintain the shape. So this is your like scaffolding, right? This is your like 2x4. They bear tension. They assist in muscle traction and cell motility, right? So motility is the flagella, weird tail thing. Actin works with other proteins called myosin. Okay, so when you see myosin and actin interactions, which we'll see questions about it. I want you in your brain to link together actin and myosin. They're both proteins and they both cause contraction of things like muscle cells. Link those two words in your brain. Actin and myosin work together to cause a contraction. Division of animal cells, a contractile ring causes the cleavage furrow. When we're talking about cell division, there's this weird kind of elastic band that is pulled around our water balloon to cause it to break into two. That's your microfilament. And then we have intermediate filaments. They're fibrous protein made up of varying subunits. They are permanent structural elements of the cells. They again maintain cell shape. They anchor things in place, and they form something called the nuclear lamina. So these are kind of like your microfilaments, right? That they kind of maintain that structure. But they are less about the motility of things and more about maintaining that permanent structure. So this is the stuff we like build our houses out of, if we're still in that kind of realm and analogy of the cell as a city, right? The nuclear lamina lines the nuclear envelope. Sorry, I don't know why my thing turned off. Yeah, the nuclear lamina is just the thing that lines the nuclear envelope. So got to make it nice and soft and squishy on the inside. All right, cool. Thanks, guys. I'll see you in class.